Hello. It is a thrill for me today to be able to tell you about some of the research in my lab that has to do with the role that neural activity plays in setting up the brain. But I thought I would start with telling you my history of doing research here at Cal. So I got my undergraduate degree in physics. I actually spent my first 2 years at UCLA and transferred to Berkeley as a junior and graduated with an AB in physics. I actually stayed at Berkeley to get my PhD in physics. I then did a postdoctoral research position at Bell Laboratories. And there I actually worked with a group of physicists who were developing tools that could be used by neurobiologists to study the brain. And this was my first introduction to neurobiology. So for my second postdoctoral position, I actually came back to UC Berkeley, where I was a Miller Fellow, and I worked with Carla Schatz, who was a card-carrying neurobiologist. So that was my first real dive into doing neurobiology research. My first independent job was at the National Institutes of Health, where I was an independent investigator. And my first academic job was as an assistant professor at UC San Diego. And 15 years ago, I came back to UC Berkeley, where I'm now a full professor. So given my long history with this campus, it is a special thrill to be speaking at a homecoming event. So what I want to talk to you about today is my research area, which has to do with how the brain gets wired up during development. And this is, of course, a very challenging process to have happen. And that's because the brain is a very complicated organ. So the brain is this organ that resides inside of your skull. It's comprised of the cells that make up the brain that are called neurons. And we think of those as individual computational units. So there's 10 to the 11 of these cells in the brain, and each one of those cells is connected to roughly 100 other cells and receives inputs from roughly 100 cells. So there's actually 10 to the 13 connections between these cells that are being made. So now the essential function of this organ is to bring in and make sense of all the sensory inputs, the smells, the sounds, the light that comes into your sensory systems. It does computations on that information, and then the output of the brain are behavioral outputs, things to move your muscles, to produce language, and lots of other internal states. So an example of this might be, say, if you're listening to a homecoming speaker on some afternoon on a nice, lovely day, the sound of their voice combined with the lunch you just had may lead to the behavioral output of sleep. So hopefully that's not happening to too many of you. Okay, so before I get into how this complexity arises during development, I just want to give you a short primer of neurobiology. And you can imagine this is what it would be like to be a student in my Bio 1A class, because this is actually a slide that I use to introduce neurobiology to the students. So, the brain is made up of these individual cells that are called neurons. Neurons are very similar to all the other cells of your body. They have a nucleus that you can see in, that's shown by the gray circle there. But the neurons also have many specializations, and that is that their membranes have these protrusions that come out, and these protrusions are called dendrites. And this is where the information comes into a neuron. And then all of that information that comes into the neuron via the dendrites is integrated by that cell body, and it all gets condensed compressed into this one cable, which is the output of the neuron. It's this wire that's referred to as an axon. So the flow of information within a neuron is input through the dendrites and output through the axon. Now that axon is going to target either another neuron, or it might target an internal organ, or it might target a muscle in your body. And when it targets that other neuron, it forms a structure that's called the synapse. And you can see in the lower left, there's a highlight of what a synapse looks like. So that's where the axon of one neuron touches the dendrites of a second neuron. And there it releases chemicals that are called neurotransmitters. And that's the basis with how neurons communicate with each other. So information flows into one neuron, into the dendrites, out through an axon, and then into the dendrites of the next neuron via these synapses. Okay, so that's your primer on neurobiology. I wanted to show you what a real retinal neuron looks, or what a real neuron looks like, and using an example from the retina that you can see in the lower right. So all that beautiful protrusions that are coming out in the back are the dendrites, and you can see that one single a wire that's going down and to the right, and that's an axon. And you can see up to the left, up to the upper right is the axon from a nearby neuron that is also sending its axon to form the optic nerve out of the retina. Okay, so I want to emphasize that development, we're interested in development, and development is a very dynamic process. And there's actually kind of a famous movie in our field that came from this lab, Elizabeth Haynes' lab at University of Wisconsin in Madison. And what you're going to look at is this is a fluorescence. It's a movie of the development of the sensory systems of a zebrafish. And so I'm just going to play the movie here. Let's see if this works. And what you're seeing are two lines of neurons that are in the spinal cord of that zebrafish. The tail is at the left end. You can see it flicking a little bit. And as this embryonic zebrafish is developing, you can see that it's elaborating this amazing set of dendrites and axons that are connecting up the nervous system so that this zebrafish will be able to make sense of the external world. And so what I'd really like to emphasize here is that when a nervous system is developing, not only are the individual neurons growing, but you can they also have to sort of find their correct targets in the rest of the body and not make too many mistakes. So it's an incredibly dynamic process. Okay, so given there is this tremendous complexity in the brain, there is some forms of organization. And one of the organizations that are found in the brain is that different processing of different sensory systems happens in different areas. So what you're looking at here is a schematic of the human brain, the parts of the human brain that mediate vision. And we refer to this as the visual system. So vision begins when light reflects off objects out there in space. So here's an example of a person looking at an apple. So light will reflect off that object. That light that's reflected off the apple gets focused onto the retina, which is this little sheet of the brain that's at the back of your eye. And so there's a lens in your eye that gives a nice image of that apple onto the retina. Then the neurons in the retina will have axons that come out of them. Their axons form the optic nerve. And that optic nerve takes all that information from the retina and it makes synapses with connections in the brain. So the first synapse that it makes in the brain is in an area called the visual thalamus. And so there it's going to touch the dendrites of neurons in the thalamus. And then those axons of those neurons in the thalamus will then make connections into visual cortex, which is the first big stop in cortex. So this is the sort of complexity that has to arise during development. And that's what we're interested in understanding. What are the signals that allow this to happen with as few errors that occur? So to give you a sense on how the visual system develops, what you're looking at is a schematic of the human visual system before birth. And so the way that the visual system develops is that the eye develops independently of the brain. And at these incredibly early stages, as shown here, 50 days gestation, that the axons that are coming out of the eye are already making connections with the brain at this incredibly early stage. Now in our lab, the experiments that we do are all in mice, and mice are actually born at a state that's much earlier in development than humans. So humans open their eyes at 5 months gestation, and by the time a human is born at 9 months, they can see pretty well. Mice are born at roughly this period of time, around 5 months of development and a little bit before that. And so by using this model system, we're able to get insights into these very early events that are happening in the visual system. Okay, so now to understand, so now we want to dive just a little bit deeper when thinking about the retina. So the retina is not just a camera, but rather it's a little piece of the brain, and we think of it as being a little piece of the brain because it extracts information on that visual scene that comes in. So the retina itself is comprised of many different neurons, and those neurons, and we talk about those neurons being organized in what's called the circuit organization. And this idea that you can have different neurons organized in a circuit is really one of the basic tenets of neurobiology. And actually, the retina was one of the first systems that the father of Ramon y Cajal, who's considered the father of neuroscience, was quite interested in. So Ramon Cajal, the way he did his research was literally by drawing the cells that make up the retina. And when you're looking at are one of the drawings that he did, that's a very famous drawing of the retina. And I actually want to say this drawing is so famous that I actually have a new graduate student in the lab who wanted to, during her sheltering in place time, has actually made her own version of the Cajal drawing that's shown here. So this is done by Karina Bystrong, who's a new graduate student in the lab. And she did a beautiful rendition of the Cajal drawing of the retina. And what you can see from this drawing is that the cells that make up the retina, they reside in different layers. They have many different shapes and many different specializations. The way that we visualize the different cells of the retina in modern times, instead of doing these beautiful drawings, is actually by using techniques that allow us to put a different fluorescent marker in the different cell types. So here's an example of the cross-section of a retina, where you can see at the very top are where the photoreceptors are. Then you have a lot of neurons in between them, and the output of the retina is at the very bottom. In order to make this easier for all of you to process in the way that we think about it in the lab, I'm going to use this cartoon version of the retina that's shown here. So just to explain what's on the left, light passes through the lens, gets focused on the retina, which is that gray sheet, which is at the back of the eye. And then actually light passes through all of that neural tissue to activate the photoreceptors, which are at the back of your eye. Now, the photoreceptors are probably the one cell type you may have heard of. They are the cells that have this specialization to absorb photons, and they take that energy that they absorb from photons, and they turn that into an electrical signal. And neurons communicate with each other through these electrical signals. So, so the photoreceptors are the input of the retina. They turn photons into the electrical signals. Processing happens that I won't go into with all these neurons that are in between that have these different acronyms that are there. And that processing converges on the output cells of the retina, which are called retinal ganglion cells, RGCs. These retinal ganglion cells have these very long axons. Their axons make up the optic nerve, and they carry all of that processed information of the visual world up to the brain. And so it's these output cells of the retina that we want. I want you to remember that term. Okay, so what does the retina look like during development? So in a newborn mouse, a mouse is born blind with their eyes closed. On the left is a little pile of about 4-day-old mice. And at this early stage of the development, the retina, the ganglion cells are separated from the photoreceptors, and so they're not responsive to light. So the photoreceptors are also immature, and so they don't very effectively turn light into the electrical signals. But more importantly, the synapse, that BP cell, that's the cell that connects the photoreceptor output down to the input of the retinal ganglion cells. And that cell has not yet formed its synapses. So these ganglion cells are mature. They've sent their axons up to the brain at a stage of development long before there's any sort of photoreceptor input. Okay, so just because the retina is blind to light, it does not mean that the retina is silent. And rather, during this early stage of development, the retina is spontaneously active. And we call that activity retinal waves. And this is the focus of research in my lab. So the way that we visualize active neurons is there's a variety of ways to measure from active neurons. You can use electrodes to measure the electrical signals directly. What we use is a method, we use that method, but what I want to talk about today is a method we use that's based on imaging. And so in order to do these experiments, in the lower left, you see that we can dissect a retina from a newborn mouse. We can place it in a chamber where it's exposed to solutions that mimic what the normal environment would be for the retina, should it still be in the animal. And then we place that chamber under the objective of a microscope, which is what is schematized right above it. And if you look at the very bottom of it, you can see a little electrode down there and a little microscope objective, and that's where there is where we put the retina for our experiments. Now, in order to make a microscope, all you need is a light source, and in this case, our light source is a pulsed infrared laser. You need a lens, which is that microscope objective. And then you need a detector. And in our case, these are called photomultiplier tubes, and they're called PMTs that are shown there. And just to show you what one of these microscopes actually looks like, this is one that was built in our lab. So there's no eyepieces. It doesn't look anything like maybe what you used in your biology class in the past, but rather we have our objective that's shown at the bottom, at the very bottom of it. We have our photomultiplier tubes on the left, and all of that other stuff is a way to focus the laser light onto our prep that would be at the bottom. I just like to point out that this microscope was actually built by an undergraduate student in the lab according to some instructions for a kit, and we had to make a lot of specializations for our particular experiments. But these are very straightforward to build, and this was actually a summer project, as I said, for a physics undergraduate student. And we build a lot of our own microscopes to do our work. Okay, so we put our retinas under this microscope. We've actually loaded our retinas with a dye, a fluorescent dye that responds to the activity of the neurons. And so when there's an increase in activity of the neuron, you're going to see the cells are going to light up. And we're going to focus our microscope at this ganglion cell layer. And so what you're looking at on the left is this, are those circles are all individual ganglion cells that are in the flattened part of the retina. And when we do that, you can see that there's this spontaneous activity that about once a minute, you see this increase in fluorescence in those cells, which we know means that those cells are active. And so that's what we call the spontaneous activity. If we now use a lower power objective, we can actually image this activity over a much bigger region of the retina. So what you're looking at here is this kind of flattened retina, but it's almost a quarter of the whole retina. So you're looking at over 1.5 square millimeters. And when you do that, and you'll see in this movie, I've changed it so that the active cells are going dark. You can see that these waves have these amazing propagation patterns. So they spontaneously initiate, they propagate for some distance, and they stop, and they continue doing this for throughout the time of our recordings, which will be for hours for our experiments. Okay, so this activity pattern is something that has fascinated me actually ever since I was a postdoctoral researcher here at Berkeley, and I was in the lab of Carla Schatz. And as I told you, I started my time in science as a physicist. And so the first thing I wanted to do was really, and while I was a postdoc, was to characterize all the propagation properties. And so we were able to learn things like these waves initiate randomly at any location on the retina. So there isn't one special site that initiates them, but they can initiate anywhere. They propagate out to just a finite extent, but then another wave will initiate in some other location. And over time, these individual retinal waves will tile the entire retina. I should say what's shown here are the summary of the retinal waves that are recorded in 4 continuous minutes of recording. The left panel is in the first minute of recording, and the little time bar at the bottom tells you when that colored wave initiated within that minute. And so you can see, although there seems to be tiling within minutes, if you look over time, the waves are constantly shifting in their shape. So when I went to start my own lab, and what I've worked on is to try really hard to understand the circuits that mediate these waves. So for and to understand what role these waves play in setting up the visual system. So I want to start by talking about some work that was done by Alex Tiriac, who's a postdoc in the lab, that gets at this question about what is the impact of retinal waves on development. So, for example, if we get rid of these waves, does anything get messed up about the visual system? And so so we've spent many years working on this, as have many other labs, and we have found there are many things about the visual system that are disrupted. And I just want to give you this one example that came from Alex's work. And that example that we're going to talk about is an organization within the retina, which is that the left eye, the inputs of the left eye remain segregated from that of the right eye. So the way that we do this is to look at this segregation of left eye and right eye through the first few synapses of the visual system. The way we look at that is we put a fluorescent dye of one color in one eye, and then a fluorescent so we'll put green in one eye and we put purple in the other eye. And that dye gets taken up by the axons of those retinal ganglion cells. And then you can see where those axons land in the brain. And it turns out that at the first synapse that they land at, which is in this DLGN, that's written there in the VLGN, that's visual thalamus, you see that the left eye and the right eye inputs actually segregate into different regions. So the way that we visualize what these axon termination zones look like is we use this method called cubic that's shown on the right. So what cubic lets you do is it's a chemical reaction that gets rid of all the lipid, and it essentially makes the brain transparent, but it also while keeping, fixing those fluorescent molecules so you can still see them. And that allows us to image through the entire brain so that we can see where the retina projects to all throughout the brain. And so then we can use a microscope that allows us to section throughout the whole brain and see what those projections look like. Okay, so here's an example of looking through that visual thalamus. And what you're looking at on the right, you can think of as the right eye or the contralateral projecting eye. And then the purple is the left eye. And you see if we can now get three-dimensional reconstructions of what these projections look like. And they're actually, the left and the right eyes are actually segregated from each other. It's a little bit easier to see that segregation if you just look at sections through the thalamus, and that's what you can see here. So if you look, say, you can see that there's a little patch or like a cylinder of those ipsilateral, we'll say right eye projections, while the green, that's right where the axons from the other eye are absent. So if you look at just one of those sections, you can see there is this segregation of the contralateral and ipsilateral eye. And this is actually at postnatal day 14, which is right when the mouse opens its eyes. They're very nicely segregated. It turns out they don't start segregated, and that when the retinal ganglion cell axons first grow into this region, they're actually intermixed. The left eye and right eye are intermixed. And so the hypothesis we wanted to test is that waves were important for driving the segregation of the left eye and the right eye. And so the way we tested that is we developed a mouse line that had altered retinal waves. It actually had very few retinal waves. And when we did that, we found that this segregation pattern was absent, right? So that and it's these sorts of experiments that we do to argue that these retinal waves are very important for driving the development of the organization in the visual system. The implications that this sort of lack of segregation would have on mice is that they have much blurrier vision than they would have had before, and that their binocular cues are not what they are in a regular mouse. So it does have implications on how well the mouse can see. Okay, so I just want to give a little intermittent summary of where we are and what I've told you so far. And that is that vision begins in the retina, this little piece of the brain that's at the back of your eye. That the connections between the retina and the brain emerge at very early stages of development before there's any visual experience of the animal. And that these retinal waves are a robust source of activity before vision is matured. And it's required for this precise connectivity to emerge in the visual system. And I have to say that most of my career was built on this idea that there's no vision, right, at these early stages of development, but rather there's just this spontaneous activity, and the spontaneous activity is important for development. But then there was a discovery that was made at UCSF by David Copenhagen, who's also a Berkeley alum. And what he found is that you can take a mouse pup that's 4 days old and it will turn away from the light. And let's see if I can get this movie to play. Yeah, so these are 5 different mouse pups. They're in little tiny chambers. And there's going to be a light that flashes on the left. Those are little LEDs. And when that light flashes, all the mice turn away from the light. Mouse pups don't like to be out in the light. This is called photoaversion. They actually, when they get exposed to light, they actually let out a call. And the idea is that means they've kind of crawled out of the nest and the mother will come pick them up and bring them back. Right, so that seems to indicate they can see. But I just told you that if you look at the retina on the right, the wiring of the retina is that the photoreceptors are not connected to the ganglion cells. So what is triggering this response? And what the Copenhagen lab showed is that there is this class of ganglion cells. These are output cells of the retina that have a certain specialization, which is that they themselves also absorb photons and get activated by photons directly. So they're called intrinsically photosensitive retinal ganglion cells. And actually they were discovered maybe about 20 years ago to be very important for mediating what's considered non-visual light behaviors, such as photoentrainment of circadian rhythms or the pupillary light reflex. So not form vision like what we use to get around in the world, but other behaviors that are triggered by light. And photoaversion is one of those behaviors where people can find light, some people in particular can find light very aversive. What the Copenhagen lab showed is that these cells turn out to be there incredibly early in development, and that if you were to make a transgenic mouse that doesn't have the photosensitivity in those cells, that the pups no longer turn away from the light. So this sort of changed everything. I can no longer say that mice are blind, these immature mice are blind, because they do have this light sensitive cell. And so this led to some research that was done by a series of students in my lab. And what I'd like to talk about today was the thesis work done by Franklin Cavall-Holm. I should tell you, both Alex and Franklin in these pictures are actually looking at the solar eclipse. So that was a big event in our lab, and you can see that they were quite excited by it. Okay, so Franklin had this question, which is that when you're looking at if a ganglion cell is stimulated by a wave, or if a ganglion cell is stimulated by light, the brain has no way of telling the difference, right? It's just firing action potentials. So how can a mouse tell the difference between that neural activity is generated by a wave, by a spontaneous event, or whether it's or when it's generated by a light flash? That was the question he wanted to ask. I do want to say that Franklin just recently graduated. This is what thesis parties look like. This is my lab during the Zoom event after Franklin defended his thesis. And that's Franklin down at the bottom. Franklin is currently a postdoc at Harvard Medical School, where he continues to study questions about these cells. Okay, so what did Franklin do? So I had showed you how we used imaging as a way to look at spontaneous activity. We also wanted to use imaging to look at evoked activity. And so in order to do this, we used the same microscope I talked about before, but now you can see there's this left half of the microscope I hadn't talked about, which is this digital mirror device, which is essentially a projector that's projecting images down onto the retina. So we can stimulate these photosensitive cells directly, and then we can look at their response. We can look at how they respond to it. And so hopefully you'll be able to see what this looks like. Let me see. So you can see very clearly when we flash the light that there's a bunch of cells that respond directly to that light. And what one of those cell responses look like is shown at the bottom. So you can see that there's a big increase in fluorescence every time we flash the light. In this particular movie, we actually blocked the waves, and so they weren't interfering with looking at the light responses. Okay, so when Franklin did these experiments, and you may have noticed that the cells that are light responsive seem to have different ways of responding to light. So the first thing that he wanted to do was to characterize how many different kinds of light responses were they. So I'm not going to go too much into the details, but this work was published by Franklin, and actually with help from an undergraduate researcher in the lab, Yizhen Zhang. Together they were able to show that, that you can just categorize the different light responsive cells into 6 groups. So what you're looking at here are these 6 different categories of cells. And what Franklin did for these experiments is he exposed them to light intensities that got bigger and bigger intensities, as you can see. So they went from very dark to to gray, and then dark to a little bit brighter, and then dark to a little bit brighter. And then he recorded the responses, and the heat maps are just ways of looking at the fluorescence changes. He used a method that's called sparse principal components analysis, which is just an unbiased way of saying, I'm just going to classify these based on their light responses. And he got these 6 functional groups. And you can see to the right that those functional groups have different responses. So if you look at the top row, that is a cell that we're going to spend a lot of time talking about that is very sensitive to very dim light. And it has a very sustained response during the whole time the light is there. If you look at the pink cells, they're very different. You need a lot brighter light to activate them, and they have a very transient response, right? So you can see that they respond very differently to light. Franklin went on to show in this study that you could actually separate these different cells by a series of different experiments that I won't go into the details. But basically, there are 6 categories, 1 through 6, and it turns out that the M1s, which are the category for the first type, are kind of cell autonomous, and they sit by themselves and they just respond to light. Doesn't matter what anyone else does. All the other cells, M2 through M6, actually form a syncytium of cells that are coupled through a structure called the gap junction. They're just all electrically coupled together. And so they all go at the same time. So even though they have different light responses, they're coupled together in this way. So this is a clue that Franklin can now use to test which of these groups of cells were important for driving this photoaversion behavior. And the way that he could do that was to take advantage of different proteins that are expressed by these cells that give them their different properties. So, for example, he can now block the light response on the M2 through M6 syncytium of cells by injecting a drug into the eye that blocks this structure called the gap junction. And then they all lose their light response. While it turns out these M1 cells have an ion channel, which is the way by which they're excited, and that's called TRPC6 and TRPC7, and we can get a mouse that's lacking TRPC6 and TRPC7. Okay, and so he wanted to see which of these cell types mediates photoaversion in these mouse pups. So the way he quantified this behavior was by putting a little triangle, exactly what you see. He taped that onto the head of the mice, and then he would turn on the light, and then he would automatically keep track of the head direction of the mouse. And what's plotted on the right is the degree to which the head turns after the light turns on. So the light turns on, and the mouse starts moving its head, and eventually it turns its head 180 degrees away from the light, indicating it's aversive to the light. He did this experiment to see how the mouse pups turn away from the light in regular mouse pups, so this is quantified by the maximum head angle. And he did it in control mice. You can see when mice are in the dark, they move their heads a little bit, but they don't necessarily turn their heads away. When you put them into a bright light, they turn their heads away. They go more than 90 degrees away to avoid the light. And that's also true at the really high light levels. But if he repeats this in the mice that lack this TRPC6/7, which means those M1 cells are now not responding to light, you can see this behavior is dramatically reduced. If he repeats that experiment in the mice, this is the data that goes into quantifying it. Its maximum head angle is a function of time. And you can see that in the dark, they don't move their heads very much. But in the black traces are control mice. They move their heads when you get to higher intensities. And when you block these gap junctions, they move their heads just as much. So blocking gap junctions did not impact photoaversion, while getting rid of that M1 cell did. So from this, he concluded that photoaversion was mediated by the M1 cell. So this particular subtype of iPRGCs mediate this photoaversion in pups. But now we can get at this question. So does this mouse, so at the same time that there are these retinal waves, why aren't the retinal waves making the mice turn around, right? Because it seems like, as far as we know until this research, all cells participate in these waves. And so therefore it seems like the waves should make the mice turn away, but really but we know that only the light makes the mice turn away. So Franklin's in the process of trying to understand how this can work. And the way what he found is that there might be a reason that these particular intrinsically photosensitive ganglion cells actually don't participate in waves. And I just want to give you an example of what his data looks like. So what you're looking at are fluorescence traces of 3 different cells. And those increases that are shown by those red dotted lines happen when there are waves. And the blue bars tell you when light stimuli came on. So these cells are not intrinsically photosensitive ganglion cells. And so they don't see the light. And they get excited by the waves. Okay, then there are some intrinsically photosensitive ganglion cells that do respond to the light. As you can see here, they have a very quick response to the light, but they also participate in the waves. But if you remember, these M1 cells are the ones that have this, they're very sensitive to low intensities of light, and they respond all throughout when the light is there. And that's the signature that you see at the bottom. And you can see they respond very reliably to light, but they don't respond to the waves. And so it looks like there's some way that this particular cell type can remove itself from the wave. Okay, and so that's how the mouse can separate waves from can separate the waves from the spontaneous from the light stimulation. Okay, I want to close with just one quick experiment that I think is really exciting that's being done by Christian Wufo, whose name I have forgotten to put on the slide, and about how waves might influence the development of iPRGCs. And we're just going to end with this movie from her, which is where she is the first person in the lab who's been able to measure waves even earlier in development, which is before the mouse is even born. And this is and you can see from these movies that there are waves that are there incredibly early in development, and that they have this tremendously rich set of spontaneous activity. And it turns out the iPRGCs are also there early in development, as indicated by this fluorescence image on the right. And so Christian has been able to show that if you look at all of the ganglion cells, they're all participating in the waves, as you can see in the orange lines. And if you look just at the iPRGCs, they are also participating in waves. And so she's currently looking to see if we eliminate these waves, what happens to the development of these iPRGCs. Okay, so to summarize what I talked about today, retinal waves are a robust source of depolarization prior to eye opening, and that occurs also from Christian's data before birth. So they're there from embryonic times up until 2 weeks after the mouse is born. That light stimulation of these intrinsically photosensitive ganglion cells mediates a behavioral response at the same age that there are retinal waves. And that the visual system may differentiate spontaneous activity from this light-evoked activity by excluding these particular subtype of iPRGCs from the waves. And the lab is currently working on trying to figure out how those cells are able to be blind to the waves. And that waves may play a role in regulating iPRGC maturation. Okay, so these are the people who do the lab. This is the lab celebrating Pi Day, which you may remember is before we had to shelter in place. And these are, I've listed the current members of the lab. In blue, I have put the people whose work I mentioned. And of course, I wanted to mention Franklin and Yijin, who have both recently graduated, and my funding comes from the National Eye Institute. Thank you all very much.